Thank you very much, Shashi. And thank you for the nice introduction and for inviting me to this winter school. I'm very excited. I think it's an exciting topic. So I think I can share my screen, right? So let's do that. Can you see my slides?
We can see your slides.
Okay. And let me start.
We can see you also next to the slides.
All good?
Yeah.
Okay,
yeah,
great. Yeah, so for this series of lectures, I want to talk about bioenergetics of self-organization, which is, I think, is a emerging and new field, which fits very nicely to the theme of this winter school. So the goal here is trying to combine basically two fields. One is bioenergetics that deal with how living systems convert energy and matter into a form usable for the system. The other aspect is how do living systems build themselves? That is self-organization, right? So I think there are a lot of tremendous work done in either parts, but I think that very new things are happening now at the interface. So that's what I actually want to introduce to you guys, and focusing more on the cytoskeleton, on the molecular and the cellular level. So for that matter, I'd like to divide my lectures into three parts. I will be giving 3 lectures this week. So the first part, I will be talking about active matter theory, a very great framework to study self-organization, and also do a few case studies applying active matter theory to the cytoskeleton self-organization. The second part is metabolism, right? Which is I will be focusing on how to measure metabolic activities with high spatial and temporal resolutions in living systems, in this case, in living cells. And number 3, the third lecture, I will be bringing you guys to the forefront of the current research by applying some of these techniques and tools that I will be introducing in lecture 1 and 2 to study biophysics of energy metabolism in development. So that's how this series of lectures will be organized. Today, I'll be talking about the first part. I'd also like to introduce our moderator and teaching assistant, Yash. So Yash is right now a graduate student at Harvard University in Dan Needleman's lab. He's my former colleague, and we're still collaborating right now. So thanks so much, Yash, for helping me out with this. I think as the lecture is going on, I would like to have some kind of interactions if possible, and I think Yash will be helping, helping me with that. And he will also be helping with the tutorial sessions, which I think we designed some very interesting interactive modules for you guys to play with. All right, so first I'd like to introduce the concept of biological self-organization. I think this is a concept I'm sure which should be familiar for most of you guys. This is basically how living systems build themselves, right? So living systems, they consume energy, and then they build themselves by assembling molecules into subcellular structures and cells into tissues, tissues into organisms, and organisms even into collective. So somehow my slides is playing automatically. I'm gonna interrupt a little bit. And how organisms also form into collective flocks or swarms of animals. So this kind of self-organization spans across a very wide length scales. And I like to quote a famous mathematician, Leibniz, who said something more than 300 years ago, that I define the organism or natural machine, a machine in which each part is a machine, whereas the parts of our artificial machines are not machines. So I think this sums up very well living systems for us. In short, it's basically saying living systems are machines made of machines. So I think this holds true even to this day, and still we know remarkably very little about those living machines. And the questions here, big questions, is like, how do molecule and the cellular components self-organize to form the cellular structures, cells, and tissues? So here I'd like to show you a movie that discusses this self-organization of cytoskeleton, which will be mostly what I will be talking about throughout this series of lectures. So let's play this movie. And there's actually a voiceover over this movie, and I want to see if you guys can hear it. If not, I think just watch the movie and I'll probably explain a little bit. So let's play that first.
The cytoskeleton is a dynamic network of filamentous proteins.
Can you hear anything?
Extends throughout the cytoplasm.
Yes, we can.
Struts, cables, and girders that give the cell its shape, internal organization, and mechanical support for movement. There are several types of protein filament, each built from subunits that rapidly come together or break apart, allowing these elaborate structures to be assembled and dismantled wherever they're required by the cell. Microtubules are stiff, hollow tubes that anchor other organelles and serve as tracks that guide the movement of vesicles and other cell components. Actin are thin, flexible filaments that form cross-linked bundles and branching networks. Actin is particularly important for cell movement. They are central to the contractile engine in muscle cells and provide the mechanical force for cell movement at the plasma membrane.
So I think this nice short movie basically displays this highly dynamic self-organization of the cytoskeleton, in this particular case, actin filaments and microtubules inside of the cell. So here, let me zoom into a little bit about those components, right? For example, the actin or the actin-myosin network, and that is this filamentous system that is very important in playing an important role in cell motility. For example, where the actin, they are basically polymers, and one of the very important processes is polymerization and depolymerization of those macromolecules from the smaller components, which are monomers of actin monomers. And this process requires energy in the form of ATP, where this monomer of actin can bind to the ATP, and then they can be added to one end of the actin polymer, and they can also be disassembled from the other end, and forming this cycle, and therefore building this filamentous structures in the cell. Another process for those actin-myosin systems is that we have this motor proteins. In this case, for example, myosin, which can bind to the actin filaments, and also using free energy from the ATP hydrolysis, they can form those power stroke cycles where the myosin are basically moving or transport the actins and sliding them, and also transport cargoes along those filaments. So this process are happening all the time in the cell. For example, a very good example is cell motility. How do a cell move? So here I'm showing kind of a schematic of a stereotypical cell movement, where when the cell is moving on a substrate, there are actins that are actually at the interface between the cell and the substrate, and also at the cortex. And they form this very dynamic networks, as you've seen in the previous movie, that they, for example, can polymerize at the front leading edge of the cell, and to form those laminipodia structures, where they form a protrusion of the cell. And then that would actually, it's like a foot that actually move a step forward, grab onto the substrate, and then the actin-myosin network will contract, for example, under the action of the motors, and then that would move the back of the cell forward, and then the cell will detach from the substrate, and then that's basically like a step forward. And if we zoom into those actin networks, as you also see in the movies, for example, showing that they are constantly polymerizing and pushing the plasma membrane forward, and that happens, for example, at the leading edge. And actually, more than just individualized active polymerization, this process is actually quite collective, and they are being regulated by many proteins, and they even form those branching structures. But collectively, they are doing this treadmilling of basically polymerizing on the front and depolymerizing the back, and then that kind of pushing the cell forward. Again, all the processes use ATP, needs energy. And here is also an electron microscopy image that I actually can show you in real life what these structures look like. It's a very dense mesh. So here are a few examples of cells moving under this type of mechanisms. For example, this is a fish keratocytes of a cell in the eye, where this is a very motile cell. They move really fast. And in this case, this is basically an experiment where you put a cell on the substrate and they move very quickly. And what I want to emphasize is that this requires a very elaborate spatial temporal dynamics of ectomyosin activities. So it's more than even just the protrusion, right, of the ectomyosin network at the forefront. They also involve the disassembly of the ectomyosin network, contraction of myosin at the back, and even this kind of hydrodynamic fluid flows of those ectomyosin networks. And different cells actually have slightly differences in organizing this ectomyosin networks to have motility. For example, the keratocytes, the fast motile cells are having this particular patterns. So therefore, this just give you an idea of this is very dynamic pattern in space and time. For example, another example here is this dictyostelium, which is a slime mold or amoeba that live in the soil. These cells also are quite motile. They can be moving individually, and they can actually form very collective aggregates if you actually see some of those very interesting social collective behaviors of those organisms. Here, there's a very nice experiment that shows that the ectomyosin, again, the ectomyosin dynamic is very important to give the cell motility to these cells, where, for example, normally the cells look like this. They have also, again, this kind of laminipodia, which are enriched the ectomyosin structures, where they basically form protrusions and they can move around. If you disassemble those ectomyosin networks using, for example, a drug called trunculin, basically you kill all the ectomyosins, you basically depolymerize all the actin filaments, and they become rounded up and they don't move. And now if you wash out the drug, you will see the recovery of the ectomyosin networks. They start from those patchy structures everywhere. Those patchy structures start to coalescence and then form those actin waves that actually sweeps through the cell, go to the edge, and now the cell become again in these irregular shapes because they've been pushed by those ectomyosin networks under this kind of wave propagation, and then they regain their motility, where you again see this kind of laminipodia forming. So the message here is that cell motility is controlled by this very elaborate spatial temporal patterns of ectomyosin networks. Okay, I want to give you another example, which is a microtubule, right? Which is another very important cytoskeleton component. Again, this system also are made of those filamentous structures of these microtubules that again can be polymerized and depolymerized, consisting of those monomers, in this case tubulin. And they do that, in this case, the tubulin can be incorporated into the microtubule using GTP. In this case, not particular ATP, it's GTP, another nucleotide, again, which is reaching free energy. And so they take, again, the advantage of the hydrolysis of ATP to extract the free energy and to allow for this polymerization to happen. So again, it takes energy. And also there are motors. In this case, there are many different types of motors. For example, kinesin is one type of motor that walks, I think in this case, towards the plus end of the microtubule. There are polar structures, and then to slide microtubules apart, for example. And the microtubules in this case are very important in cell division because, for example, they form this structure called spindle that segregates chromosomes. Again, I like to show you a movie of demonstrating cell division and the spindle formation in real time, in real life. So this is a slightly sad cell, I think. Sometimes cell division could be a little bit strange. I can see the chromosomes condense. and then the spindle forms and it segregates the chromosomes into two daughter cells. This is mitosis, and this is how cells proliferate. Okay, so this basically should give you an idea of this very dynamic structure of spindle, right? That forms during this mitosis, and if you look inside of this spindle, they are made of microtubules. For example, in the mitosis, they can be nucleated at the centrosome, for example, and they will actually be connected to the chromosomes. And actually, it's more complex than this. The nucleation is happening all over the place throughout the spindles. And there are also a lot of motor proteins that are moving around, like the kinesin I've shown before. They're also dyning, other motors, and they are acting collectively to slide microtubules apart and to generate the force, for example, eventually to segregate the chromosomes. I have to say that actually, the dynamic of spindle is still under very extensive research, right? I think in cell biology textbooks, you guys know, the spindle, there are segregated chromosomes, but how do microtubules nucleate? How do this process happen from a physics perspective? It's still under very intense research. So I think it's also a very exciting field. For example, here, people now can do EM, electron microscopy. And this is also doing electron microscopy and on a mitotic cell. And under that, you can actually reconstruct the whole mitotic spindle and actually can count every single individual microtubules. For example, and this is a very painstaking work, for example, from this group here, also in Dresden. And if you successfully segment it and reconstruct it, a mitotic spindle can count. For example, in this case, there are 4,884 microtubules inside of this one spindle. And microtubules are for the different categories. They are the ones that are connected to the chromosomes through the kinetochore. And those are kinetochore microtubules. You can even segment those because you can literally see which microtubule is connected to the chromosome. And here you have about 792 microtubules that connect to kinetochores, for example. And they have different stabilities. So, all right, this is to give you kind of a fast crash course on the cell cytoskeletons. By the way, I want to emphasize that if anybody have any questions, feel free to ask. For short questions, we can discuss. Feel free to interrupt me and we can discuss along the talk. But there will be also one or two discussion parts that people can actually interact more thoroughly. So now, for today's talk, I would like to give you this framework, right, to present the framework to study this type of collective behaviors in the cytoskeleton, and that is using the active matter theory. So, okay, Yash, I think I saw you raising hands.
No, I think he was telling that other people can raise their hands by raising his hands as an example.
Okay, all right, good. Okay, so, okay, here I think active matter. Okay, I want to do a simple survey here. So how many people in the audience have heard of active matter? Could Yash help me do a little bit of survey? Like half, more than half, for example?
More than half.
More than half, okay. How many people have worked in the field of active matter physics? Do we have I cannot see the room too clearly in terms of the.
Yeah, no, that's about 6 of them.
Okay, good. So I think that's very good. A lot of people have heard of it, but I think, some people have worked on it. So I think it's a great time for me to go also a little bit in details about the theory. So active matter, right, is defined as living materials composed of self-driven units or active particles that each are capable of converting stored or ambient free energy into systematic movement. Okay, and this is actually definition coming out of this very nice review paper from Marchetti and al. In 2013, and also recommend as a very nice reading for you guys. So, okay, the keywords here are active particles that self-driven, right? And then they are able of converting energy into motion, right? So that basically means, for example, a very simple example is some self-propelled particles, right? So in contrast to passive systems like gas, for example, in this room, which are not self-driven, right? Here, the particles dissipate energy at a local scale, at the individual particle scales, thinking about a microtubule, thinking about a cell, right? And they are self-driven. And then they can also collectively interact to form those collective behaviors, which are very rich in dynamics and is absent in the equilibrium counterparts. For example, here people have done this very amazing in vitro experiments of mixing microtubules with motors. And now motors are driving each microtubules, and they're self-propelled systems or particles, if you will. And then you put them all together, they form this very beautiful, elaborate patterns, right? Do the same thing for acting. You also see very interesting patterns, even including those type of motile waves, which I will talk about a little bit more in detail. But this to show you that just with this very simple conceptual idea, putting a bunch of self-propelled particles, you can capture a very amazing rich behaviors of spatial temporal dynamics of active systems. But the question is, like, how do activities and interactions lead to such rich dynamics, right? Once you strip to this kind of active matter system to its core, it's easy to start to model and study them. So, for example, in the next few slides, I will introduce some kind of modeling approaches here. So one part is kind of particle-based simulation, right? And this is actually pioneered by VSAC, right, in 1995, where they first proposed kind of this physics way of studying the collective behaviors of the self-propelled particles. Okay, so here in this talk, I will be throwing out some equations and some formal theories here, but I will try to make it intuitive, so you don't have to understand all the gory details, just get the concepts. So, for example, you can model each individual self-propelled particles by writing down equations of motions, right? So in this case, you can model its position, and the change of position is basically related to its cell propulsion. So the key is all in this V0, in this cell propulsion velocity. And that's the active term, right? That's how the cells are dissipate energy locally. So they basically move along a certain direction, parameterized by this angle theta. Each particle does that. But they can, and also the angles are also can be evolved. In this case, they interact with each other. So in the VSAC model, is dealing particularly with a bunch of active particles that are polar. They have a propelled velocity, they can move themselves, and they have direction, right? They have a director. So this direction is subject to interaction with its neighbors. So if you have a bunch of other neighbors around it, which are moving in certain directions, they want to be aligned. That's the simple term. So there's alignment strength that basically a particle will be looking at its neighbors and compute its kind of average neighbor orientation and try to be aligned with it. This process is always subject to noise. Like in biology, there's a lot of noise, so you have to fight that. It's also a noisy term. So simply with the self-propulsion, alignment, noise, you get this fundamentals of active matter system. Okay, so here you can do this through computer simulations, right? You can start to initiate a bunch of particles. They're polar, they move along their own direction, parameterized by theta, by this angle, and then they look at their neighbors and they try to compute the average direction and they move together. Okay, so very simple. And you can actually do numerical simulations. I think Yash, during the tutorial sessions, will help you guys run through some of those practical simulations. And so you get an actual feel for the system. Okay, so here, but what's fun about this system, right? So you put a bunch of particles and they interact and they move by themselves and they align to each other. But then what do we gain from it? This is what you get, right? So a useful physical systems is you gain more than what you just put in, right? So here you gain a lot of interesting collective behaviors that is hard to predict just by looking at single particles. For example, there are three characteristic phases for the simple model. One is everybody moving together in the same direction. It's a polar phase, thinking about like a bird flock, for example. And then there's a disordered phase. Apparently, if noise really overwhelms the alignment or the density is really low, everybody just doing their own thing. Somewhere in the middle, very close to this transition. So there's actually a phase transition, and disorder phase transition. Very close to this transition, you have this actually coexistent phase of moving waves in this disordered background, which is very highly non-trivial, right? So people have spent decades actually studying this very simple system and figure out phase behaviors and figure out the nature of the phase transitions, right? This is also kind of reminiscent of how people study phase transitions in gas and liquids, but now with active particles. So now we know that there are very interesting behaviors in this type of systems. How does it relate to experiments? Okay, so in this case, again, let's go back to our cytoskeletons. So people have done very interesting experiments. Here I'm citing this work from 2010 from Shatter et al. They put actin filaments that basically stabilized, I think in this case, stabilized and polymerized actin filaments. They put them on, and then they put a lot of mouse motors grafted on a cover slip. So there are motors living on the cover slip. They throw this actin filaments on top of it and just add ATP to it. And then the motors basically bind to the actin filaments and start moving them around. And then they can tune the density. They can tune the density of the actin filaments and see what happens, right? So if you look at the model, it has a prediction, right? So if you increase the density, you would go from a disordered phase into an ordered phase, right? Or you decrease the noise, which is definitely more difficult to tune. So you can tune density, right? So you can actually see to test the type of model predictions. So first, if you have a very low density, you have just a few actin filaments, right? On this in the system, you have a disordered phase. Okay, and now if you increase the density, let's see what happens. Right, so this is at intermediate density. You increase the density, actually above a certain threshold, and this type of moving clusters forms. The clusters of those actin filaments. Okay, so then, so again, right, you can see this clusters movement. And then if you keep increasing the density, well, and then this is what happens. So you go to this phase where you have this kind of a propagating wave, right, moving through this kind of more or less disorder state. All right, so again, zoom in, you can see this in detail. All right, so now here's the phase diagram. All right, if you have a very low density, you are in a disordered phase. Above a certain critical density, which can also be predicted from, for example, from the VSEC model, you enter into polarized state. And in this case, of course, the reality is always more complicated. You actually have this kind of homogeneous clusters, and then you gradually move into this kind of density wave phase, right? So it's a very rich dynamical behaviors, but in essence, right, this kind of order phase transition and this type of propagating wave phases, they're all being predicted with a simple self-prepared particle model. So, and then let's after the VSEC model, right, in 1995, in 1998, so Toner and Tu started to also think about this kind of active systems, but from another approach. Rather than this kind of particle-based simulations, they're thinking about continuum models or field theories, right? And this is reminiscent of, hydrodynamics, right? How do we study, fluid? We're not modeling, for the most part, individual molecules, but we just look at fields, right? Velocity fields, density field, etc. So they did the same thing. So now we can define a density field, right? That basically describes how, the local densities of the particles. You can then write down a continuity equation for density fields, right? Where the partial temporal derivative of the density field is basically related to this convection, right? So because they're active particles, so they basically, if you have a certain direction, and then they basically move along that direction, and that convects their mass. This is basically conservation of mass. And also, you have this polarity field, right? So, like I said, there are particles that have internal degrees of, they have this internal degrees of freedom, if you call them, which is this local, polarity, right? So internal polarity. So they have a preferred direction of motion. So for that, you introduce a polarity field that basically tells you, if you think about locally, you average a bunch of particles, and what is kind of the average, sort of a direction of motion, or their polarity is, that's characterized by a vector, right? In this case, which would be related to both the magnitude of those polarity. 0 is completely disordered. 1, for example, is ordered. It depends on how you define it. And then it also gives direction, which is a director field. Okay, so you can write down another equation for this polarization field, right? Which, if you look at this, I think probably would be a little bit reminiscent of a Navier-Stokes equation, right? But there's some distinctions. First, there's this kind of highly nonlinear term here, which is due to this alignment, right? So this part would basically dictate this disorder transition. And this is due to this kind of alignment interactions, which is usually like absent, for example, in this Navier-Stokes equations. And then, of course, you also have this kind of viscous damping, which actually is not too different in the Navier-Stokes equation. You have a pressure term, but here the pressure term is kind of dictated by also this kind of activity. Okay, so this is V0, again, can consider that as the cell propulsion speed, which is the active term. All right, so now we can, instead of running those kind of particle simulations for thousands or millions of particles, you write down 2 fields and you describe the system on a hydrodynamic sort of scale. So the advantage of this is you can reduce the degrees of freedom, which makes it amenable to analytical analysis. So Toner and 2 basically wrote down those equations in 1998 out of just symmetry arguments. And then people since have been studying these systems also quite intensively, and they both run particle-based simulations, and also they run the numerical solutions for those field equations, basically through, let's say, finite difference schemes. And then, for example, they can match the phase space, and even the continuum theories, or the field theories, can also capture a lot of the very interesting phase behaviors, even include this type of traveling bands in this disordered phase, really close to the disorder phase transition, or hiding non-trivial states. So this basically means a continuum model and particle-based model, they can be connected. And so here, the point of this part, I think I want you guys to be able to know the tools that you can use, and actually even want to give you guys a little bit of ideas of how are these 2 descriptions, particle-based model and field theories, are connected. So the particle-based model, for example, in this particular case, they have 3 model parameters, right? Cell propulsion, alignment, and noise. And okay, depends on very specific kinetic models. You can write down slightly different variations of this, but the essence is just like this. There are 3 parameters, but there are thousands or hundreds of thousands of degrees of freedom, depends on how many particles you put in your system. Well, continuity equations, you actually have more parameters if you just write down the equations by symmetry arguments. In this case, for example, there are actually over 5 parameters, because you don't know what those A and B's are. There are some nonlinear terms that are gonna give you these order phase transitions. You don't know what the parameters are, so you actually have more parameters. But of course, you have much less degrees of freedom in this case, which you just have this 2 fields. Well, but now, what if we want to get the best of both worlds, right? So can we constrain the parameters of the continuum model? If we kind of say now we are going directly from a specific kinetic model, and let's see if we can actually derive those continuum equations and connect them explicitly. Okay, so this is usually a quite technical part, but I don't want to go into the technicalities. I just want to show an example, right? Like what can be achieved. So in this case, for example, okay, so what do we gain, right? We can reduce the number of independent parameters in the continuum equations by connecting them explicitly to the specific particle-based kinetic model. So all those 5 unknown parameters, is there any way to make them a functions of those specific kinetic parameters in a specific kinetic model? Can we do that? The answer is yes. And if you do that, you probably need to go through quite elaborate mathematics, but the concept is very simple. You define those fields, density fields and polarity fields, explicitly, right, related them to the local averages of the particle degrees of freedom, and then you define a relevant field. In this case, for example, density field, of course, it comes out naturally. Polarization field, I just described that. But here, actually, to make the mathematics simpler, you actually can actually come up with this field, W fields, if you will. Basically, it's like a product of density field and polarization. So in other words, the more dense there is, naturally, you have a higher magnitude of this field. If you now replace the polarity field with this W field, you can actually derive very explicitly those field equations of density and this kind of, let's say, redefined polarity, where all the model parameters, right, all the model parameters here, which used to be A and B and V0, we don't know what they are, now becomes explicit functions of the kinetic models. Okay, so now this continuum model literally have the exact same model parameters as the particle-based model, and then it's amenable to analytical analysis and everything. So I think this is where this type of very systematic coarse graining, which I call coarse graining, where can do to us. And I think this concept, coarse graining, I kind of want to emphasize this in this series of lectures, is a very useful technique in physics, where you have a very complex system, but somehow if you define the right parameters and you do this local sort of averaging, you can actually get to a description of the system at a higher level that are much simpler and can also preserve a lot of the collective behaviors at large length scales. Okay, so if you want to know more about this type of derivations, you can also go into literatures, for example, here. All right, so, okay, let me just, for example, give you an example like, okay, now we get this field equations, but what do we gain? Okay, thing is, now you can analyze it much more elegantly, right? So if you want to know, okay, how do I know if there's any actual disorder order phase transition? Very simple. You can even take the activity to 0, right? So not an active system anymore. Get all the V0s, right, which is self-proportional, to 0. So you get rid of all the terms that have V0. Now you'll be left with this very simple equations, right? And now you say, I'm at steady state, right? So all the temporal derivatives go to 0. And you can solve for this very simply. You can literally take a pen and pencil right now, and you can actually derive this, where first you would have a homogeneous density field, right, from the first equation. And then you can predict there will be two different phases in terms of the polarization field. One is disorder, right? This field is 0. That happens if your noise term overwhelms the alignment term, right? And actually overwhelms the alignment term in the sense that it overwhelms the product of alignment strength and density, right? So this also predicts that this kind of phase transition is density dependent. And also, if you crank up the alignment strength, or you crank up the density of the system, now you are going to be driven into this polarized state, where you actually will, okay, Yash?
There's a question, Xingbo. In the second equation, on the second box on the right-hand side,
yeah,
the equation for W, I think you're missing a W there.
Oh, yeah, sorry, I'm sorry. Yeah. Exactly, okay, you got me there. There's a W right here.
Right. And the other argument point was that if you have V naught equal to 0, the advection term disappears. Yeah. But the del square W does not in general disappear, but this might be an artifact of the particular kinetic model that you're looking at. Yeah, you're right, exactly. I think in this case, I do not have this rotational diffusivity. I think, let me put no, I think I do have the rotational diffusivity, that's just a noise term. Exactly, I think it's a particular case of this type of coarse graining. And in general, this term does not have to vanish. You're right. So this, I think, is more to give you a very conceptual understanding in this case. Thanks for spotting there should be a W vector right here. But this is to give you a conceptual understanding that under this particular coarse graining scheme, and if you actually also neglect some high order terms, which I also did not talk in detail, and then you can actually simply discuss this order phase transition. And then, for example, and in the ordered phase, and also in this case, actually, to answer that question, you can also say, what if I'm in a homogeneous state, right? In that case, actually, all the grads would also go away. So I'm kind of also discussing this kind of homogeneous state in that sense. Maybe it's more accurate to describe that. And then you can actually compute this, this kind of homogeneous polarized state
that can predict its particular, right, a magnitude of polarity as a function of noise, density, and alignment strength. And this happens when your density or alignment overwhelms noise, right? So this kind of gives you a flavor of how you can, right, so use those field theories to predict those phase transitions. If you add back the cell propulsion, you got a zoo of patterns, which I will be describing next. Okay, so at this point, I think it's a good place to pause. And if anybody have any questions, because next I'm going to go into some case studies. So I'd like to see if there's any questions. All clear?
Hey, Xingbo.
Hey.
Maybe I'll ask a question, maybe that'll inspire more people. So if one to think about energy consumption in these things, in these particle models or continuum models, we learned about, being ATP, giving free energy to things to do work. Where would we put that in those models that you described?
Yeah. It's a very good question. For example, right, this is actually what is missing, right, in active matter theory, where people know the system dissipates energy locally, and they're completely far from equilibrium. And for example, and then they put this activity, in this particular case, just in the cell-propelled velocity, right, V0, right? So in a very simple case, right, I think this, again, it's an open question. I kind of want to start up discussions, in the field. You can, for example, put V0 as a function of this energy input, right? Then you have to talk about what is the right energetic variables in there. So that's a one possibility, right? And of course, this is only one class of active matter model that I'm proposing, the cell-propelled particle model. There are many other different type of models that probably all more explicitly take into account of active stress or forces, right? In that case, that probably can depend on the activity or the energy input, because we know motors, for example, they use ATP. Yeah.
Thank you.
Yeah.
Any other? Yeah, we have a question here.
Go ahead.
Hi. So in the motility assay video that you showed of the actin polymers, what it seemed like was that as you kept on increasing the density, in the beginning phases, they tended to sort of cluster together, and later they filled the entire field. The one before this.
So for here is a cluster.
Yeah. So before the density was increased, like to sort of an extreme, they tended to cluster together rather than being sort of spread out homogeneously. So why exactly is that happening?
Yeah, very good question. Okay, so I don't have an exact answer for this particular system, but actually, in the case study, I also show you there are cases when some type of phase separation happens, right? Where you have a dense phase and less dense phase. So, and actually, I will be introducing that even without this type of alignment. You can see actually there's some simple mechanisms that allow you to have this, phase separation, right? In terms of dense phase and dilute phase. So I think stay tuned for the next part. If you still have questions, right, there will be another discussion parts.
Yeah, there's one more question. You can...
Yeah.
Yeah, I'm referring to the equation which looks like the Navier-Stokes equation. So in the alignment term, is this like, have we chosen it to be represented as a linear, as a quadratic equation? Can we go to higher orders? And what do A and B signify here when we have chosen this kind of an?
So in this case, I think this type of okay. So in terms of the order, the quadratic, for example, if you can first get this through let's say kind of a Landau-Ginzburg type of free energy, right? So in that case, you can actually, it's kind of a very classic type of first order phase transition. Another way to get this is if you go through this explicit coarse graining, right? Explicit coarse graining. That's what it's describing here. And you also get this particular quadratic form explicitly if you have a particular type of alignment interaction. I think this term in general, I think it could be even higher order terms or different orders. Depends on, I guess, the nature of those type of interactions. But I think this quadratic form captures, like I said, this one of this a particular class of kinetic models, and also can follow from some type of this Landau-Ginzburg theories. Okay, so I think that's one question. What's the other one? I think you asked two, right?
Did you have another question?
Oh, I think the previous student asked also about A and B. Sorry, can you repeat that question?
Yeah, what does A and B signify here?
Oh, yeah, so A and B, for example, here, they are just logical parameters, right? So, again, but here, I think the better way to answer this is that if you actually, let's say, coarse grain, right, a particular kinetic model, you will see very explicitly what A and B could be related to. So I introduce you to look at this one. So now, A and B, this makes it A now, and that's B, right? So A and B, in this case, basically depends on this type of noise, as well as the alignment strength, right? So, so I guess in this case, if you just look at this field theory, this would predict a certain phase transition, right? A disorder and order if A and B follow certain relations, right? But what A and B could depend on is, for example, in this kind of particular model, is noise, interaction, and density. And that actually predicts precisely that it's noise or density or alignment strengths that attune could change A and B, and then that could lead to this disorder order of phase transitions.
And there's one more question here.
Yeah.
So in this model, if I got it right, you are assuming that the interaction is a metric kind of interaction. So there is a sort of radius around a given point, and you count, you take everything within that radius. So would it be different if you rather consider the topological one? That is, you count the number of neighbors no matter how distant they are.
Exactly. Yeah, that's an amazing question. I think there are actually some work exploring this in the context of information transfer, like in bird flocks. I forgot the exact arguments of that papers, but I think there are ways to actually distinguish even these two different interactions, metric or non-metric. I think their argument is, in William Bialik's paper, I think they also showed that even this topological type of interactions are also quite relevant, like in bird flocking, I think, if I'm not mistaken. But I forgot about exact model predictions there, but it's a very good point. And regarding this model, for example, for coarse grain, for the sake of mathematical derivations, here, if you go through the derivation, you would actually kind of make the interaction radius like very, very small, almost like within delta function. And then you can kind of proceed. You can, I think, to explore those, like if you sort of have a kind of very kinetic specific model, and then maybe, you put in metric interactions or put in different interaction with different, put in topological interactions or metric interaction with different, radius of interaction, and you can probably explore some of the consequences like that, even through simulations or through this field theories. Yeah.
Yes, one more question, Xingbo.
Yeah.
I had a very basic biology question. Like you talked about low density being more to like a disordered phase. I just wanted to know how disordered in general biological systems are, and do they like the disordered, like we also talk about collective motion and all those, so they're mostly an ordered phase, right? So how much of this disorder or this decrease, like low density, actually contributes or has a significance in biological systems?
Yeah, very good question. Yes, so disordered phase. Okay, so the first thing that can come into my mind is, which I will show you a kind of a case study of, for example, of cytoskeletons of let's say, both ectomyosins and spindles. For example, I will go into detail there, but just give you a flavor. I think during this kind of pattern formation, right, the ectomyosin networks, they can be, for example, they have very, elaborate spatial-temporal dynamics, and they can go through different density phases, right? For example, a spindle, right? They will be assembled first from just microtubules, right? They have been nucleated, and then they actually gonna interact and form the spindle. So through the whole process, it's likely, it's possible that would also undergo some of those kind of order transitions, depend on, how their kind of density and or the kind of interaction profiles are being regulated throughout those process, including, for example, the example I'm showing, cell motility, where when you wash out, right, the ectomyosin networks in the dicty, and then they recover, right, then they start to renucleate, right, into the spots, and then it sort of coalescence and form waves, which is not too, right, unlike a potential sort of a disordered order phase transition. And actually, maybe this is not a good point for me to go into those case studies to show you that some of those phonological models might be even relevant for biological systems in terms of those phase transitions and collective behaviors. And this will also probably answer the previous question about why there's like actually a dense clustering in the dilute background, for example. Okay, so I guess maybe now let me just move on to this case studies. All right, so now I will show two case studies. One is for ectomyosin networks in the dicty, right, which is motivated by this very elaborate pattern formation of ectomyosin networks, where if you depolymerize all the actin networks, they're gonna repolymerize, right, and then they form this kind of spots, patches, and then they become this kind of waves, right? So for example, you can speculate, would there be any actually order phase transitions right here, right, as density grows? And then the waves, right, will reach to the edge of the cells, and then they can become motile again, right? So this is a highly dynamic processes, and what are the minimum mechanisms to generate those patterns, right? So here I'm doing a phonological approach, right? So, okay, if you zoom in, you can actually see that at the cortex, there's a very dense mesh of ectomyosin networks. So what I presented right before in this kind of hydrodynamic model, even the particle models, or mostly the hydrodynamic models, is that we are more or less in a kind of dilute limit, right? Where the particles are still more or less, right, independent, except for, okay, they want to align with each other, but they're kind of not getting in each other's way, right? So, because if they do, you can imagine they might impede each other's motilities, right? So you would not be able to describe the system with a single constant cell proportion speed, right? They might be dependent on the local environment. Okay, so this type of local crowding is possible, right, to generate this type of impediment on their cell proportion. If that happens, okay, I'm going to use the same framework, right, of the hydrodynamic equation that actually derived, right, from this particular kinetic model, where I'm introducing this polarity field that's conjugated to the density. And here, a very simple, again, phenomenological thing you can do is you assume that your cell proportion now is not a constant, but it depends on local densities, right? If you do that, you have to move your velocity, right, into the gradient terms now, because it depends on density, which is the field. And if you do that, again, you have to also consider this polarity field. And here, I'm actually gonna even make it simpler that I'm not considering alignment anymore, right? I just say alignment is 0, and now you're dominated by this noise terms. If that's the case, this is a very fast variable that's gonna relax very quickly to 0, right? You only don't have alignment, you only have noise. You're gonna relax to 0. So for that, before I do that, actually, I also want to introduce the density-dependent velocity. Okay, that would make this the other velocity go into the gradient term. And then you say the polarization relaxes very quickly because there's no alignment anymore. And then you can eliminate this polarity field, and you can write down a single equation for density fields, where that would have this form where you basically have a density-dependent diffusivity, right? Because of the diffusivity, if that is a constant, you move it out. It's basically just a diffusion equation. But here, because of this density-dependent velocity, now you have a density-dependent diffusivity. And then if you were to do a little bit of a stability analysis, you ask the question, is a homogeneous state? So this probably would answer the question proposed by the other student before, like why do you have dense field versus dilute field? If you now ask, is the homogeneous field in terms of density homogeneity, is that field stable? Okay, now if you do a stability analysis, you would see it would not be stable if this effective diffusivity now changes sign, which is possible now because it involves this kind of derivative of this cell proportion, which now is density dependent, and this term can change sign. So once the so this basically predicts if your density is above a certain threshold, and then you would, right, your homogeneous state will be unstable, and you will have a spontaneous phase separation. Okay, this is a very, I think, beautiful prediction. If you want to know more details, you can actually refer to this paper, but I want you to show you a simulation. Right? I'm going back to simulations now, particle models now. Right? So this is the back and forth. It's also how you can sort of go to the continuum theory, make some predictions, and do your numerical experiments and to see if the prediction is correct. Right? So now I just predicted if density is above a certain threshold, just put a bunch of cell-proper particles, even no alignment direction, just let them bounce into each other. What's going to happen? Right? So here is a beautiful simulation. All right, they are sweeping this parameter of density and cell proportion. Okay, so look at this. Above a certain density threshold, you form this phase separation. You have dense regions, right, in the background of disordered regions. They're not motile anymore because there's no polarity, there's no alignment. Well, so this is being termed as the motility-induced phase separation. And this happens without attractive interactions, right? So which is actually a very surprising result that because you can be reminded of the liquid, sort of the gas-liquid phase transition, and then requires the attractive interactions, for example. This does not require that, but you would have this phase separation. So this is kind of shows you just by putting in this activity, right? You'll be able to generate patterns that is right, not possible, right, in its equilibrium counterpart if you don't have an attraction. Okay, so okay. By the way, I have to know how well I'm doing in time. Okay, I still have I think 30 minutes. Okay, so here now, so we just being able to recapitulate a sort of spontaneous phase separation, right, in this active matter model by just putting cell proportion with some crowding effect. Okay. Now, in this actual myosin system, for example, which is what we're trying to capture its dynamics, that the density is not conserved, right? So because there's this polymerization, depolarization happening, right? So we're talking about those polymers, so they can basically, be born and die, right? So the conservation of mass is actually broken. So in order to model their dynamics more accurately, we actually have to go further, than this this crowding effect, where you basically say, now I can add this logistic term, right? I can basically add a growth or death or polymerization, depolarization term into my equations of, in this case, just density. So in addition to the first term, now I just put certain growth rate here, right? And then postulate a certain logistic function here. If you do that, you can show that in addition to this phase separation, which you have a giant dense phase and dilute phase, now you can actually have this patchy patterns now, right? Where this basically logistic term, together with this effective diffusivity, is setting a characteristic length scale in your system, right? So this is to show you how just a simple equation looks almost like diffusion, but diffusivity is density dependent, and brick conservation of mass can basically, generate very interesting patterns. And this can go also into this literature, which describes this type of generic route to pattern formation, might be able just to describe, let's say, bacterial pattern formation, and in this case, also potentially active mouse networks. So All right, so this is basically a second ingredient, right? Break the conservation of mass. Now, that's basically, more or less resembles these patchy patterns, right? Again, I'm doing a more morphological type of study. But what about waves, right? Because these patch patterns, they have to coalesce and then they have to go to the edge, they form waves. And then, until then, you get motility. So what are the minimum mechanisms to generate these waves? All right, so for that, we now, right, remember we disregarded the polarity field. Let's just add that back under the argument that now the alignment strength is probably pretty strong. So this polarity field would not decay that fast. So it becomes also slow variable, comparable to the density fields. And if you do that, you just add alignment into the picture. Now, in addition to these patchy patterns, you gain internal structures of those clusters, which they can become, right? First, if you have very low alignment, okay, then you have those very isotropic basically states, right? They're basically isotropic clusters. But if you increase alignment, the cluster starts to form structures and eventually they become waves, right? So become propagating waves and they have it in this phase separated state. So this is to basically show you that just with very simple phenomenological rules, you can actually capture a very rich phase space of pattern formation, right? I'm using this ectomyosin as a motivation, right? I'm not saying this is exactly what's happening in this dictee, but this gives you a tool, right, to study things from top down. Okay, but now I'm going to give you an example that's more related to experiments, right? How do this active matter theory, how do we know that they're like right, in a sense that they can be really quantitatively compared with experiments. So then let's go to spindle again in this case. So again, spindle is also like actin-mouse networks, it's highly dynamic, right? Again, it's also polymerization, depolymerization, and there is also this motor activities where the kinesin, for example, is sliding microtubules apart, right? They generate active stress in this case. All right, so with all those processes happening inside the spindle, among many others, the spindle is fluctuating, right? Even if you have a steady state spindle, its components are not static, right? They are constantly fluctuating in density, polarization, and active stresses because of this local activities happening. Actually, because of the local input of energy, right? Going back to the theme of our talk, of this winter school. So all those activities are driving these active fluctuations. So can we measure those active fluctuations? And can we, from those active fluctuations, infer some properties of the spindle? Okay, so this is actually a work done by my colleague here, Jan Bruges, when he was also a postdoc in Dan Needleman's lab. And they did this amazing work where they basically measure those active fluctuations. For example, one thing they can do is they can measure this director field. In this case, this director field is basically the polarity field, right? Except now you normalize it. So it goes from 0 to 1. And you can actually directly visualize or measure this field from what is called polarization microscopy by taking advantage of this biofringence properties of microtubules. I'm not going into the technical details, but just know that you can measure the polarity here. And then density, right, another density field that we are dealing with, then you can just label turbulence, and then you can actually measure the density field. Okay, again, the same spindle here. Sorry, also the same type of spindle here. And then, right, so now how do we apply our theory to the system to learn interesting informations? Again, you can write down your polarization, equation for polarization field or director field, right? In this particular case, so this is slightly different class of active matter compared to the self-propelled type of active matter that I'm proposing, but essentially they're the similar thing. So I'm not going to go into details what are the actual differences. In this case, for example, they have this director field. First, of course, they have this local elasticity because they are just filaments and they actually, like liquid crystals, for example, they have elasticities. And then, because this is an active system, there are actually flows inside of them. There are also flows that are coupled to this, a polarity field. And the density field, which is kind of similar to what I just showed you before, there is this type of convection terms, and also there's a logistic terms because of polarization, depolymerization, and also there's this type of transport and diffusion. Okay, so continuum equations and describe those active matters now apply to spindle. So, but how do you actually apply to spindle? Like, what do you learn from this, right? So, cool thing is you can actually quantify now the active fluctuations. For example, you can measure at each point this director field, right, through this pulse scope, where you can actually look at its fluctuation around a certain mean, right? So, for example, this delta n, and this n average is the mean polarity, and this delta n is the fluctuations around the mean. And now what you can do is you can do a autocorrelation study, right? Autocorrelation of this the director fluctuations around its mean. You do that in the Fourier space, the technical details, where you can actually predict how the fluctuations behave in space and time as a function of model parameters. In this case, they predict that, right, your polarity fluctuations is dictated mostly by this elasticity constant. And then if you just feed this functional form to your data, where you actually measure those fluctuations, right? And then you can actually infer the elastic constant of this material, right? So it's one example. The other one, do the same thing with density, with density fields, where you also can predict, measure the density fluctuations around the local mean, and do a spatial temporal correlation of your density fluctuations. This is a slightly more complex term because you involve a lot of polymerization, depolymerization, and transport, but essentially it's just a function of model parameters that you can feed again to a direct model experimental measurements, and then you basically can infer again all the relevant parameters like microtubule transport flux with absolute units here, microtubule turnover rate, that's basically depolymerization rate, and the diffusivity. And the cool thing is actually, for example, Jean Brugis and Needleman, they also compared those numbers with single molecule measurements in vitro, and they agree, right? So this is actually kind of a power of this active matter theory, in this case, in application to real systems. But one more thing is that there are active fluctuations, or you can even look at just the steady state, values of those polarities, and for example, and you can actually use the active matter theory to predict the morphology or the local mean polarization of the spindle, and that agrees perfectly with experiments if you just impose a simple boundary condition where your microtubules have to be aligned right with the edge, and there are like two local defects at the poles, and then without any feeding, you can basically predict the polarity field inside of the spindle accurately. All right, so this is another message. All right, so, okay, I guess I just showed you two examples. One more from logical, that how do you, get pattern formations in this kind of active matter theories from kind of a top down, right, from logical modeling approach. The other is applying active matter theory to actually real systems, in this case, spindle, and to be able to get relevant information from active fluctuation measurements. So I think now I want to pause here because I'm pretty much at the end of my talk, and we probably still have 50 minutes here that I want to, have a little bit of discussion here. Like, what do we learn from active matter theory? What did we learn today, and what is missing in active matter theory? Also put a few just questions here just to seed the discussion. So I think feel free to ask questions and or propose your ideas. So maybe Yash can help me moderate a bit.
Yeah, sure. We already have a few hands.
Am I audible? Yeah. So what decides the direction of motion of individual biological entities? So like there is shape asymmetry or any chemical reaction happening out there. So what decides in which direction the cytoskeleton or anything will move?
What determines the direction? That's a very, very good question. Okay. Right. So okay. So I first have to admit that I'm not an expert in the exact molecular biology of the cytoskeleton. So maybe let me give you another example, which is in maybe another length scale, right? For example, for like bacteria, chemotaxis. right? So for chemotaxis, right, for example, the gradient of chemoattractant, right, that can potentially dictate the direction of motion, right? And that's actually one of the classic areas where the active matter, this kind of cell-propelled particle model is being applied to, which is this type of round and tumble motions where, for example, E. coli can do chemotaxis, right? And that's the direction is kind of favorable in moving up the gradients of the chemoattractant. Inside of the cells, for ectomyosin and spindles, there are a lot of proteins that are actually nucleating the filaments. For example, okay, I think I showed you here. So here I can just help together, like speculate together with you guys, but I would encourage you guys also to dive into some of those literatures for ectomyosin and just, microtubules if you want to know the answer or state of the art. For example, here, for this type of laminopodia, the active filaments are sort of moving at the membrane, cell membrane. There's also proteins that nucleate them at that edge. And then there also this, for example, this ARP23 complex, which are branching the actins. And these are probably related to how they are localized and how they be potentially, determine the direction. And also there's also kind of simple steric interactions, right? That's also a strong prediction from the active matter theory, where if you have a bunch of, active particles, which are just rods, right? And they, just from an entropic perspective even, they're preferred to be aligned, right? So that means if a density above a certain threshold, they'll prefer to be aligned, right? So that also would determine the direction. In the case of, spindles, first, spindles are nucleated, right, from the signals emitting from, for example, from the chromosomes, right? It's RAN-GTP signals. So they'll be sort of aligned, sort of those will be nucleated there. Also, they'll be nucleated at the centrosomes as well. And then they would be connected to chromosomes, and then inside of the spindle, right, and they will be polarized and also partly attributed to, some of the motor proteins and some of them just steric repulsions, and then that would determine the sort of the direction, along the spindle axis. I hope that kind of answers some of the questions.
Hey, Xingbo.
Yeah.
Sorry to interrupt, but since I have the advantage of a board over here, I'm going to take a stab at this answer just to draw a sketch. I think you are using your hands, but I will just use the board and perhaps it helps a little bit. Let's see. You're asking, how does direction come about in any active particle in general? In biological systems, yeah? So let's take, I'll give you two examples. One, let's say a bacterium, like Xingbo was saying, right, initially. So there's a head and there's a flagella, right? So the flagella can beat only at one end, and this will dictate the direction of the motion over here. Okay, that's fairly obvious where you have a head and a tail. In these examples that Xingbo has talked about, where you have these microtubules and motors, let's take the example of one of the assays that he mentioned. So normally in the cell, you have a microtubule and you have what's called a plus end and a minus end, okay, in the microtubule, meaning that there is some structural asymmetry in the microtubule itself, okay? Now, if a motor sits on this structural asymmetry will guide it in one direction, okay? In the experiment that Xingbo showed, these motor proteins were fixed on a glass substrate, and these microtubules were put on top. Now imagine that something is walking against another object, the road, but the road is the one that's free, not you. And therefore this object will move.
Microtubule or that motor?
So the motor is what is consuming energy, and it is pushing against the microtubule like that, right? But because I'm fixed, it's imagine that, there's a rope and a lot of us are standing over here and each of us is just tugging on the rope, but none of us are going. The rope will move, correct? That's exactly what's going on over there. And in the spindle, you're imagining that network of these microtubules, and you have many motors in between them, right? So each of us has ropes, and we are passing along, and the ropes are going to move between us, right? Roughly that. Thank you.
Yeah. So very good. Thanks so much. Thanks so much.
I just had the board, Xingbo, so it helped a little bit.
I know. I would say for also the future lectures, feel free to jump in to help me out.
Sorry, I didn't.
I only have my hands here, so.
Yeah. No, I just saw you struggling with your hands, so
I know.
I thought this may be.
Well, hands are good approximations of microtubules.
That's right. Yeah.
Okay, so all right. So maybe I can take one more question, and then I can actually introduce like 1 or 2 slides just to motivate my next lecture. So, one more questions?
Hi. So in one of the slides, you showed us like a resemblance to diffusion equation. I wanted to ask that when you have such confined space, like the scales of cell, the diffusion constant itself will depend on like how large the space is, how constrained the space is. Now, if this is an active matter system, there is self-propulsion. So there are 2 effects, like if a particle has to move, there are 2 effects which are playing a role. If one is diffusion, one is this self-propulsion. So which one is dominant, and how does this dominance change if the cell size increases?
So, okay. Let me try to interpret the questions here. I think you also mentioned about a kind of confinement in here, right? And then you're also asking about is self-propulsion or diffusion dominant? Do I understand correctly?
Yes.
Okay. So, all right. Very good point. So I think first, okay. Let me go to the self-propulsion diffusion. So, okay. So if you look at my equations, actually, I did not put in explicit diffusion. So I think if you look at this equations, right? So, So I did not put in explicit diffusion. System they just diffuse self-propulsion. But actually, you could have actually a diffusion terms here, right? Which is just simply like thermal diffusion. In general, for these active matter systems, those terms are the thermal diffusion is like negligible usually in the face of self-propulsion. Self-propulsion, in other way, like you can. Okay, so here you're talking about self-propulsion, which are also interrupted by occasional, right, like noise, right? Because here we're talking about self-propulsion, but sometimes you can actually have noise to orient your direction. That's what's happening, right? That's what the noise is about. And you have self-propulsion. And that over the long time scale is basically like a diffusion, long time scale, right? So your effective diffusivity is basically, if you will, this kind of self-propulsion speed squared over this noise, I think. And then that term is generally much larger than your thermal diffusion. So that's why I neglect it here. Okay. And then I think let's go back to this confinement term, right? So the confinement, if you have some local confinement, there are actually been a lot of studies where people actually put those active systems in a confined box, right? And then, of course, depends on this kind of effective diffusivity, or in other words, you can also think about kind of persistent length, right? Like how long you can run before you kind of turn direction, and how that's compared with the length scale of the box, and then it determines whether the separate particles would see the boundary or not. For example, if you are quite persistent, that also means your effective diffusivity at the long time scale is quite large, right? Quite persistent, and then you feel the boundary, right? There will be a phenomenon of like accumulating at the boundary, right? That effect would happen. And so that's actually a lot of interesting work has been done in active matter particles in confinement. So I would advocate you guys to look into those literatures. But of course, if now your noise increases tremendously and now your effective diffusivity is really become low, and then that becomes very like indistinguishable in a way to a thermal Brownian particle, and then the confinement in that case would not change much of the behavior in the bulk. I hope that answers the question. Does it?
Yes. Thank you.
Okay, good. All right, so, okay, since, because of the constraint of time, since I only have 4 minutes now, I think I want to very briefly, make contact with the theme of this winter school, it's energetics, right? Because so far you haven't been hearing me talking about actual energetics. I'm just hinting at it because active matter system that, but they by definition, right, are living materials composed of self-driven units, and they're active particles, and each capable of converting stored or ambient free energy into systematic motion, right? So the keyword here is free energy, which also been dissipated on the single particle level, right? So that's the characteristics. So we studied about dynamics, and the field has studied about dynamics of active matter, but really, like Yash was asking, how do you put energy into the picture explicitly? So, well, this is where energetics, so energetics comes into play. And we can be more ambitious. Let's do it in the cell. If you do it in the cell, then we're talking about metabolism. So I think many speakers here have already touched on the metabolism. So I'm not going into details of metabolism. We'll go a little bit into the next lecture. But here, the idea is it's like, how do we couple active matter theory to cell metabolism conceptually? And okay, so this is basically kind of a little to prime you guys for the next lecture, is that we can study this in a cell because we can measure metabolism with high enough spatial temporal resolution through imaging, right? And we can study image to spindles, for example. And here you're looking at a mouse oocyte or an egg. That's a steady state, so it's a perfect system. Just have a spindle living there, and then you can have mitochondria all around, and can study their interactions. And to show you that this is right, there is an interaction. I'm just gonna do a simple experiments, right, which is experiments I've done when I was in Needleman's lab, and which I'm continuing pursuing here in my independent research group, is I can drop oxygen, right, which is basically what is needed for cell respiration. I can drop oxygen. Of course, I can recover it, so it's see if it's reversible, and then I'm gonna image my spindle in the cell, right? So here, I am dropping oxygen very slowly, looking at spindle response here. It's a low oxygen, and it's recovered. All right, so maybe one more time. Okay, here I'm dropping oxygen for the cell. Here I'm recover oxygen. Okay, so this basically shows there is some non-trivial interactions between metabolism and active matter or self-organization of spindle that can presumably be used to bridge active matter to cell metabolism. And okay, so I will discuss some of those in the next lecture. But again, still, this is really, really much like ongoing work. I don't know the answer, but I think it would be great to use as a motivation to sort of achieve our goals here, which is like, how do we incorporate metabolism into active matter theory? So kind of, I want to motivate a lot of thoughts, right, in this winter school by showing some of those examples. And so today's TA sessions, I think, so Yash will take charge and run through some of those simulation techniques of the self-propelled particles, and also see if you guys can actually incorporate some energetics into the picture. So I would leave this to Yash, and I hope you have a wonderful tutorial session. And I think I'm pretty good on time now. So that concludes my talk. And if you have any questions, I'm still happy to answer. Thanks.
There's one question here from Surit.
Yeah.
Hi, Xingbo.
Hey.
Hey. So the spindle, you said, I didn't quite catch what's happening in that movie, the spindle oxygen drop.
Yeah.
So are you saying that it's disassembling?
Yeah, okay, it's a mystery, right? That's what I'm trying to set out to figure out. But if you look at the face value, it seems it's become smaller.
So so when you drop oxygen, it's only the spindle which is affected and nothing else?
Oh, okay. So I will show you, of course, also in the next lectures, the mitochondria definitely impacted.
Okay, sure.
Yeah. Exactly, but stay tuned for that. Yeah.
Any other questions, final questions for Xingbo's first lecture?
Hi, just a question from the same video that you're showing right now. So, I understand that this is really new, but have you looked at how much have you looked into respiration in these cases? Like how does the different parameters of respiration, let's say states and all, how do they change?
Yes, perfect question. I'm gonna dedicate an entire lecture for that. So So, and I will show you like how do we measure respiration and how do we measure that actually with spatial resolution. And I will show you how that changes. I don't want to spoil you anything much, but stay tuned for the next 2 lectures.
Just a technical question. In the video that you are showing where you're dropping and recovering the oxygen, what is being used to label the mitochondrial segments? What's the fluorescent probe, or how are you imaging it?
To mitochondria, you said, or spindle?
Because you can see the brightness go up and down, right? And
the spindle.
Oh, yeah. You're asking what I'm using to label the spindle?
Yes, yeah. What's being used to label the tubulin?
The tubulin. That's just tubulin, a dye. Right, so a tubulin dye that binds to tubulin, and that's how I visualize it.
Only adheres to the fibrils and not the monomers, that dye?
That's a good question. I think that, I have to double check if that dye binds to monomers as well, but I think the dominant signal should come from the polymers.
Thank you.
Yeah.
More questions for Xingbo? Metabolism, spindle.
Hey, Xingbo, we don't see any more questions right now.
All right.
But if there are some, we will collect them and pass them on to you so that you may address it tomorrow.
Yeah, it'd be great.
Okay. Thank you so much. Thank you so much for your lecture. Yeah.
No problem. All right. Cool. See you guys next time.